Hello, I'm Sophie Rovner from the American Chemical Society. Welcome to this news briefing from the ACS Fall 2020 virtual meeting and expo. We're joined today by Dr. Laura Fabris from Rutgers University. She's studying viral outbreaks in single cells, which could reveal new ways to defeat them. Dr. Fabris.
Good morning. Thank you very much for having me here today. So I'll be very happy to introduce you briefly our work today. So there's a couple of slides that I'll go through. Don't worry, there's not many. If you can bring the first one in, the previous. Our work focuses on using surface enhancement spectroscopy, or SERS, to understand and to identify viral RNA in individual cells that are intact, that have not been lysed, and to study their evolution. What is important to understand is that this approach can be adapted to various viruses, various RNA viruses, but in this particular case, we looked at the influenza A virus. The influenza A virus is a RNA virus, and is composed of 8 segments. Each segment, as you can see here on the left, we focus in particular on segment 4, the hemagglutinin segment, is composed of conserved regions that are marked by the blue bars here. There are a few hundreds of nucleotides, whereas the center bases are exposed to a lot of mutations as the virus replicates. The way we adapted our approach to identify the presence of viral RNA in individual intact cells, we took these nanoparticles that you see in the black and white diagram on the right. We call them gold nanostars for their unique shape. And we functionalize them with DNA that is designed to fold and unfold in the presence of absence of the target RNA. When the target RNA is presented to the nanoparticles, the DNA folds into what we call a beacon loop and brings a fluorophore dye that is attached to the external outermost part of the DNA close to the surface. And this gives a SERS signal. This SERS signal turns on in the presence of the virus and off in the absence. We have studied the virus in individual intact cells. We distribute them in the cytosol where also the virus is present, and we determine whether the virus is inside a cell or not, and what its concentration is. So if you can go to the next slide, I'll show you a little bit how the signals look like. You see in the on signal, you see various peaks in the spectrum that we show, whereas in the absence of virus, we have only almost flat background signal present. And so we can switch on and off this signal depending on the presence of absence of virus. And the particles, each of these particles is sensitive to the presence of a virus. So you can imagine that each of them spreading through a cell can tell us whether there is a virus going around or not. The uniqueness of these particles is that we can adapt them to identifying basically any viral RNA. And so, for instance, one important target, as we know, could be the SARS-CoV-2 virus that is giving us so much trouble these days. The interesting part of this approach is that it can be extended to develop the diagnostic tests. So what we are working on right now is to implement these nanoparticles to create a sensitive and accurate diagnostic assay to detect viral RNA in circulation, and an assay that is also rapid and much faster than currently available and much cheaper. So we're currently also collaborating with Hamamatsu Photonics for integrating one of their portable Raman spectrometer for use in the clinic. A spectrometer is a very small footprint, very light, very low cost, that can be integrated with our particles to give a very rapid diagnostic assay. And if you can move to the next slide, the next final slide. And the interesting part also is that we can adapt our nanoparticle to not only give SERS signal, but also fluorescence signal. You may know that in biology labs, virology lab, clinical labs, fluorescence is the technique that is mostly used. So we realized that we have to integrate that technique to sort of blend with the existing technologies. And so we have also this beacon DNA folding and unfolding, giving a fluorescent signal rather than SERS. But also we have other design of nanoparticles, such as fluorescent nanoflares, to identify viruses. And in collaboration with the Professor Connie Chang group at Montana State University, my postdoc, Dr. Hao Wang, is currently implementing these nanoparticles in microfluidic devices where we can enclose a single cell into an individual drop, introduce nanoparticles in the droplets, and really identify the presence and amount of viruses within each drop. And so by modulating the conditions in each drop, we can tune the cells to respond in different ways to the presence of the virus. And this will be very interesting in the future to identify the presence of what we call super spreaders that are important for, because they really produce lots of viruses at rich replication, but also can allow us to predict the onset of new emerging pathogens that could have pandemic potential. And thank you.
All right, thank you very much. At this time, I would like to invite questions from viewers, and please state your name and affiliation in the chat window when you type in your question. I will get started with a few here. First, do scientists currently think that SARS-CoV-2 is mutating as quickly as influenza A or HIV?
So, well, I'm not a virologist, but, and so, you know, all the details, I'll leave that to my more esteemed colleagues. But what I know is that there is evidence because of some of the characteristics of the SARS-CoV-2 virus, it is actually not mutating as faster as the influenza virus, for instance, because of, properties of the polymerase, which is the one that actually is the enzyme that grows the RNA strand in the virus. So yes, it is mutating, but luckily for us, not as fast as the influenza A virus.
Why is it important to study viral mutations in single cells rather than in a whole organism or a population?
Well, obviously there are a lot of excellent studies done in population, but what we believe and what we have discovered over the past 5 years is that there is evidence that there is small numbers of cells that produce very large number of viruses at each replication. And so we can define them as super spreaders. Super spreaders are very important because if we identify them, we can separate them and we isolate them and stop them from, spreading the virus even further. So studying single cells allows us to really isolate these unique outliers and separate them.
What types of characteristics in a virus or a person might make that virus or person a super spreader?
This is exactly a great question. This is exactly what we're trying to understand. We are trying to understand why some cells or some people are super spreaders. What are their characteristics that may make them super spreaders? And importantly, we need to identify what are the particular conditions, environmental conditions, that will make them super spreaders. So they may be super spreading in one situation, but not in another. So that is very important for us to study.
Have any viral mutations for any virus been identified in super spreaders?
Well, it's important to understand that there are mutations occurring in any virus. Every time a virus replicates, produces mutations. But it's mostly important for us to understand rather the opposite. What does make a super spreader, rather than, and it produces a specific virus rather than a specific virus with specific mutation making a person a super spreader. But it's still a big field. It's expanding, and there's a lot of questions to be answered.
Is it known whether in super spreaders, all or many of their infected cells carry the same viral mutations that allow them to produce large amounts of virus? Or is it only a small subpopulation that have the super spreader phenotype?
So whether or not super spreaders carry the same viral mutations, it's unclear, or at least, I am not very familiar with that. But what I know is that there are only few cells or people that are responsible for they have this phenotype that makes them super spreaders. So that's why it's important to isolate them and identify them, because of their very, very small number, but huge production of viruses.
How common is it for viruses to mutate to a less infectious form? How could knowledge of these mutations be used to prevent or treat viral illnesses?
Well, it's important to understand whether mutations happen constantly in viruses, right? Every replication produces a mutation. However, it's important to know that not each mutation is productive. And often, meaning that the virus may produce a mutation, but not necessarily would make it more pathogenic, because talking from the perspective of the virus, it wants to be more pathogenic or more infectious, right? So it's not easy a priori on a new virus to understand what are the mutations that are going to be productive for the virus or productive for us, meaning they're going to be less dangerous. And often the studies are understood a posteriori, after the mutations have been isolated. And so this is why studying the phylogeny of the virus is also very important. That's what my virologists are doing. That's this very important work.
Which version of your assay for detecting viral mutations do you think is more promising, the Sars or the fluorescent assay?
Well, we are going on both fronts right now. The Sars assay is more sensitive. So it might be more useful for detecting viral presence at early onset. So when a person is just being infected, there's few copy numbers of virus in a person. The fluorescence, as I said, is more amenable to adaptation to the clinic. And so there's pros and cons in both. So far, we are carrying them both along. But I think we are very excited about both of them. And we're trying to pair them up with isothermal amplification, which compared to PCR is much shorter time, produces large number of copies. And so these would make the assay very, very fast and very sensitive. So we're very excited about this.
Do you have any sense how long it might be before Sars is a clinically approved technology?
Well, I've been working on trying to push this Sars in the clinic for about a decade. And I've seen that in the recent years, mostly in other fields, mostly more in oncology field, but in the recent, let's say 3-4 years, I've seen a lot of progress. There's a lot of technology progress, making the technology cheaper. A lot of clinical partnerships that are making the technique more spread into hospitals and making clinicians and biologists more aware of it. So, it's a steady implementation, and there are very, very many things to consider, but it's very promising and we're very excited.
Do you think either version of your assay would be practical for diagnosing viruses such as SARS-CoV-2 or influenza, in addition to studying viral mutations?
Oh, definitely. Definitely, yeah. And that's why we are interested in both. The diagnostics is important for the clinical part. The studying viral evolution is important to understand how the virus behaves and predict new viruses. So both, obviously the viral evolution has a slower pace because it's more fundamental study than the diagnostic, but they're both very important.
So you mentioned in the press release that you were collaborating with a company to develop a portable Raman spectrometer. Yes. How close is that to becoming a practical marketable device?
Oh, it's very close. Actually, the company has asked us, Hamamatsu has asked us to partner to actually validate the instrument with, some experiments. And we have been working with them for, over 2-3 years now. So we have a very good partnership, and, it's very close. And actually they've been introducing it to some, technical shows. So, yeah.
All right, wonderful. Well, thank you so much. The archived version of this session will soon be posted at www.acs.org/fall2020 press conferences. Please join us for our next press conference at 10 a.m. today on 3D printing of greener buildings using local soil. Thank you.
Thank you.